Good day, everyone. My name is Philomin Juliana, and I am a wheat breeder at CIMMYT, Mexico. And today I'll be talking about some of the retrospective analysis that we did of CIMMYT strategies to achieve genetic gain for grain yield and our perspectives on integrating genomic selection for grain yield and bread wheat. And to begin with, I'm just going to show a very brief outline of the yield testing strategies at CIMMYT and genotype resources. So this is the current grain yield testing scheme at CIMMYT. So in the first stage of yield testing, or the year one, we have about 9,000 lines, greater than 9,000 lines evaluated in the full irrigation under the bled planting system in Obregon, Mexico. And in the second year, about 1,092 lines that are selected for high grain yield and for stem rust are evaluated in 6 different environments: the full irrigation, bed planting, and flat planting systems. And then we have evaluation for drought stress in a moderate drought stress environment with some reduced irrigation. And then under a severe drought stress with just on flat beds with drip irrigation. And then we have the heat stress evaluations where we have both early zone heat stress and terminal and late zone heat stress. And selections from these, about 280 lines, then go into the stage 3 or the year 3 of yield testing in Obregon, under 3 different environments: just the full irrigation bed planting, the drought stress flat planting with drip irrigation, and then the late zone heat stress with the bed planting. And simultaneously, parallelly, a set of lines, about 540 lines, are sent to 5 different sites in India, Pakistan, and Bangladesh as part of the USAID project, which are known as the South Asia Bread Wheat Genomic Production Yield Trials. And they are also evaluated under a flat planting system in these locations. And using all the data from all these 3 years and also the target environments, the South Asia bread wheat trials, we have several different trials at international nurseries that CIMMYT distributes to different countries every year, which includes the elite spring wheat yield trials and the high rainfall wheat yield trials, both of which are targeted for the irrigated environments, and then the semi-arid wheat yield trials, or SAWITs, which are targeted to the drought-stressed environments, and the high temperature wheat yield trials, which are again targeted to the heat-stressed environments. And these are 50 lines that most of our partners receive from CIMMYT every year. And throughout the past several cycles from 2013 to 2020, more than 70,000 CIMMYT wheat breeding lines, which are in the stage 1 of yield testing, have been genotyped as part of the USAID Feed the Future project and the Delivering Genetic Gain in Wheat project. So now we have a large amount of data, and as you can see, the diversity here, again, this is showing the principal component 1 versus 2 for this huge panel. There's lots of relatedness between the parents because of some of the common parents that are used across cycles, but there's huge diversity and symmetry on plasm. So now we have the genomic data for all these lines, and we have used it to answer different questions, which are here. So the first question that we asked is there a shared genetic basis between grain yields evaluated in Obregon and the target environments? Because you need to have some similarity in the genomic regions in Obregon and different environments. And when we did a genome-wide association study for grain yield in Mexico, grain yield in India, and other international sites, we did see that the 2NS translocation, which is very interesting, which is associated with stripe rust, the YR17 gene, wheat blast, and other lodging tolerance, some other traits as well, was very consistently associated with grain yield in several different international locations as well as in Obregon. And there were other small regions, again, which were also small effect regions, which were also associated with grain yield in these different locations. And that was very interesting in understanding the genetic basis and understand that there is some common genetic basis of grain yield across these locations. And the next thing we did is pick out some of the consistent grain yield associated markers. So we have used about 47 markers associated with grain yield. And on the x-axis, we have different elite spring wheat yield trials. So that is the year when they were distributed. So it's from 2013 to 2017. And then on the y-axis, we have the frequency of alleles increasing grain yield. So those are the grain yield favorable alleles. And each line here represents the favorable alleles for a particular marker on that chromosome. And as you can see, in most of the SWIT nurseries, you can see that the number of lines carrying these favorable alleles have increased. And then you can see most of these are like close to fixation, especially the 2NS region, where you can see from 2010 onwards, you can see a very sharp increase in lines carrying the 2NS translocation. So that was a very good indication that selection and phenotypic selection for grain yield has worked, and then we have been able to increase the frequencies of favorable alleles by phenotypic selection for grain yields. And now with markers, some of these could be really pushed much faster. We also looked at what is the grain yield performance of the SWIT lines in contrast to the local check. And for this, we used 12 different sites where we had a good amount of data from the 24th to 37th SWIT. And as you can see in this figure, these are the 12 different sites that we looked at this. And the red lines here, or red dots here, show an SWIT line with the highest yield in that particular SWIT in that particular location, and the yellow lines show the yield of the local check. And as you can see from this figure very clearly, about 95% or 144 out of these 152 site year combinations, there was a very clear superiority of the highest yielding SWIT line over the local check in these locations, which is very encouraging. Despite you selecting in the selection environments of Oregon, you still have a very good impact in the target environments. And the next thing we wanted to compare was look at the genetic correlations between the environments in the stages 1 and 2 of yield testing in Oregon, and 3 sites in India where the South Asia bred wheat genomic yield trials were evaluated. So that's Jabalpur, Ludhiana, and Pusa in India. And you can see 4 different cycles, 2014 to 15 to 2017 to 18. And each one of these panels represent the Oregon environments, the selection environments, and they are the stage 1 full irrigation bed planting, stage 2 full irrigation bed planting, stage 2 full irrigation flat planting, stage 2 reduced irrigation, stage 2 drought stress environment, and stage 2 early and late zone heat stresses. And as you can see, the genetic correlations between the Oregon environments and the South sites in India where the subwoods were evaluated, where about 540 lines in each year were evaluated, are quite high. And sometimes you can see, especially for Ludhiana, you have a good correlation of 0.5, and then you can see again Ludhiana has a good correlation with the full irrigation bed planting 0.6. And except for these 2 heat stress trial environments, which were not meant for these irrigated environments, you can see a good correlation with the other environments in Oregon and in South Asia and in India. And sometimes you can also see that even for a site across the different years, there's quite a range in correlations. Genetic correlations, one year could be 0.3 and the other year they could be 0.56. So that we also observe that range in genetic correlations because of the high year effect. And the last 2 things I'm going to talk about today are the perspectives of integrating genomic selection for grain yield. The first thing here is about the grain yield prediction accuracies in Oregon. So we did a prediction of all the different environments that I just mentioned for the different cycles. So I have 5 different cycles here. And on the top of this panel, we have the genomic prediction accuracies, and the bottom panel has the pedigree prediction accuracies. And in the, as you can see for the different years in these different panels, you can see there's not a high variation between genomic and pedigree-based prediction accuracies. And this is very consistent across all the environments and all the different cycles. There's very little advantage we gain by using genomic relationships over pedigree relationships. And we wanted to understand why this was the case. So we started looking at the full SIB family sizes and the number of full SIBs in these stage 1, 2, and 3 yield testing stages. And then if you look at this, you can see in stage 1 of yield testing, the largest family had 65 full SIBs. In stage 2, it had 44 full SIBs. In stage 3, there were just 11 full SIBs. So as selection goes, and you do not co-select SIBs in these advanced generations, so we just have very few SIBs in these generations. And a major conclusion out of that was we bring programs to just have a good pedigree and family structures similar to that of CIMMYT in the yield testing stages can just use the available pedigree, which is free of cost, versus using genomic relationships, because in these generations, you're not going to see much advantage of using genomic relationships or the pedigree. And this has to be done much earlier in the cycle where you have a large family size. The last thing we did was looking at prediction accuracies for grain yield in the South Asia bred wheat genomic prediction yield trials, which had about 540 lines. And I'm just showing you the number of lines with some missing data here. And they were predicted from the yield of the same lines in the selection environments of Oregon. And as you can see here, so you can see the stage 2 different environments. And the first panel here is showing the accuracies from the EL model, or the environment and line effects model. So this model has no genomic effect, no G by E, nothing. It just has an effect of the environment and the effect of the line. And the second panel here shows the effect, the environment, genomic, and genotype by interaction effect. So that's the G by E model. And then the third panel is just showing the phenotypic correlations between the Obregon environment, and Jabalpur, Ludhiana, and Pusa. And then we have 4 different cycles that we tested this for. And as you can see very clearly in this figure, the accuracies using the environment line model and the G by E model were very similar in most cases. And so they were also very similar to the phenotypic correlations. And a major conclusion here was that if you were to predict 2 different environments, the prediction accuracy is going to be equal to your phenotypic correlation, and the accuracies, or it's going to be very similar to your phenotypic correlation, and the accuracies that you get from just an environment line model are going to be very similar to a model from a G by E, especially if you're just predicting the same set of lines in 2 different environments. So at all times, G by E models are not going to be very useful for genomic prediction. So that was our take home from there. And the overall conclusion from this was genomic selection for grain yield is still very challenging because the trait is highly influenced by the environment, and multi-environmental trials are still indispensable for reducing risk and to continuously achieve genetic gain for grain yield. So with that, I would like to thank the Bill and Melinda Gates Foundation, the USAID, the Foreign Commonwealth and Development Office, and BGRI, Kansas State and Cornell University, all of which are involved in this project. And thank you very much.